Hi there! Today, we are going to talk about bacterial conjugation. Bacterial conjugation is a transfer of DNA by direct cell-to-cell contact. Recall your knowledge about plasmids. Plasmids are small double-stranded DNA molecules that can exist independently of host chromosomes. Also recall episomes. Episomes are plasmids that can exist either with or without being inside the host's chromosome participation. Conjugation involves the participation of a very important plasmid called the F factor. The F factor contains 28 genes located in its TRA operon. It's very important to note that the F factor can exist outside the bacterial chromosomes or be integrated into it. There are three types of bacterial conjugations that will be discussed. The first type of bacterial conjugation is the F plus by the F minus mating. Gene transfer is unidirectional, meaning there is a definite donor, which is F plus, or the fertile cell that contains the F factor, and the recipient, the F minus, which is the non-fertile cell and does not contain the F factor. An F plus strain contains an extra chromosomal F factor just lying around the cytoplasm. This F factor contains genes for the formation of the sex pilus for plasmid transfer. This sex pilus is used to make contact between the F plus cell and the F minus cell. The pilus brings the F minus cell closer to the F plus cell, and when close enough, the F plus cell prepares for gene transfer by assembling a type 4 secretion apparatus from the sex pilus. The F factor inside replicates by a process called rolling circle replication. During rolling circle replication, one strand of the F factor is cut by a complex of proteins called the relaxosome, which are encoded by the F factor. The site of which where the relaxosome cuts the F factor is called the ORIT, or the origin of transfer. The strand of the F factor is cut, it is being cut into two parts. The first part is called an F prime part, and the other part is called the 3' end. Relaxase, an enzyme associated with relaxosome, attaches itself to the F prime end. The 5' end of the F factor goes inside of the F minus cell. Once inside, the 5' end goes into a circle formation and replicates. As the F factor replicates and the 5' end goes inside the F minus cell, it passes through the type 4 secretion apparatus system. The entering strand is copied inside the F minus cell to produce a double-stranded DNA. Once complete, the F minus cell becomes an F plus cell containing an extra chromosomal F factor, just like the F plus cell from before. The second type of bacterial conjugation is the HFR conjugation. Some cells in the population have their F factors integrated into their chromosomes. These cells make use of this type of conjugation, and the donor in HFR conjugation is called the HFR strain. The plasmid can still operate and direct the sex pilus, carry out rolling circle replication, and transfer genetic material into the F minus cell. DNA transfer starts when the F factor is cut into two parts, mainly the 5' end and the 3' end. Again, the process remains the same like that in the F plus by the F minus mating. The only difference is that initially, only a part of the F factor is transferred into the F minus cell, followed by the chromosome. Note that the connection between the cells break down, so not all of the F factor is transferred. Thus, the recipient remains an F minus and does not change into an F plus cell. The third type of bacterial conjugation is called the F prime conjugation. Because the F factor is an episome, it can separate itself from the bacterial chromosome. Sometimes, when this happens, it takes with it a small piece of the bacterial chromosome. The newly plasmid formed is larger and more genetically distinct from that of a moderate F factor. So now, it's called an F prime plasmid. An F prime cell, a cell containing the F prime plasmid, can only mate with an F minus cell, but take note that their mating is very similar to that of the F plus by the F minus mating. The F minus cell becomes an F prime cell and also becomes partially diploid due to the presence of a bacterial chromosome in the original F prime plasmid. Now you might be asking yourself, what is the difference between an HFR conjugation and an F prime conjugation? Well, the answer lies on the plasmid used. In the HFR conjugation, the F factor is integrated into the bacterial chromosome, while in the F prime conjugation, the bacterial chromosome is what is inside the F factor due to the F factor separating itself and taking a small piece of the bacterial chromosome when it separates with it, thus forming the F prime plasmid. That concludes this short video about bacterial conjugation. We hope you learned something, and don't forget to share, like, and put a comment down in the comment section.